Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture 53, where I will discuss flavonoid, flavones, isoflavonoids and proanthocyanidins in brief. So, and in the subsequent class I will bring the issue of several interesting metabolic engineering applications, including the anthocyanin pathway. So, in this class first I will make an overview of different flavonoid route, then flavone and isoflavonoids, then proanthocyanidins and orones. These are the things I am going to cover now. So, biosynthetic pathway leading to the formation of flavonoids. So, here this is a brief overview of this. So, as is the chalcones. So, chalcone is basically formed by joining malonyl CoA, 3 molecules of malonyl CoA with 1 molecules of fumaroyl CoA, or the same joining can leads to the formation of stilbene, which I have already covered in the previous class, and stilbenes accumulated into grapes. Okay. So, these chalcones depends on the plant species, where these chalcones can lead to the formation of isoflavones and isoflavonoids, or these chalcones can lead to the formation of anthocyanins, or this can take part in the formation of flavonols. So, here where it plays important role, or in formation of say isoflavones and isoflavonoids, particularly what we find in case of legumes. So, their chalcone synthase and chalcone reductase will work together, and eventually the pathway moves in this direction. So, what is here, that first chalcone will be converted into isoflavonones. So, and then this step is called isomerization. Isomerization is there in both the cases, and then flavanone will be converted into flavone, and it is stored as such, that is the dehydration, or flavonone can be reduced to flavone 4-ol, which forms flavone 4-ols, and flavone 4-ols undergoes polymerization and forms flovapane. Flovapane is a typical complex phenolic compounds, which accumulates and forms the maroon color, and you will find in the sorghum inflorescence, in musa flower, the spathe. Yeah, so there you will find this. So that is flovapane, is a very unique compound, but important thing is that flavanone undergoes hydroxylation, which makes dihydroflavonol, and dihydroflavonol can take part in the formation of flavonols, or it will be subsequently reduced and makes flavone 3,4-diols, which was called leucoanthocyanidin. Leucoanthocyanidins are colorless, that may undergo reduction and form anthocyanidin, which eventually forms anthocyanin, which are the colored components, or these flavone 3,4-diols will convert it into flavone 3-ols, which are the catechins and epicatechins, components of green tea polyphenols, and which eventually joins together and form the condensed tannins. So, these are the different routes which originate from the chalcone. So, either it produces flavonols, or it produces flavones, or it produces anthocyanins, or it produces isoflavonoids, depends on the different plants. But normally the flavonols are present in most of the plants, but the content may be very lower amount to get it detected. So, the first one is the chalcone, which we have studied then chalcone is formed by a chalcone synthesis. So, next one is the flavanone. So, particularly there is an isomerase I have shown the isomerization here. So, this is basically the isomerase enzyme which converts into flavanone. So, flavanone makes flavone. So, flavanone makes flavone, here it is flavone, or flavanone can makes isoflavone, or flavanone can makes flavonol. All this I have shown, this is can make flavonols, or it can make anthocyanidins. So, flavanones are also very important molecules, and through different enzymatic route it makes different phenolic compounds. Now, these are all basically derived from C6C3. So, this comes under the flavonoid. So, we can also tell that polyphenols, because more than one phenyl ring is there. So, the reactions leading to different flavonoid group, first is the chalcone, which is formed by joining malonyl CoA and coumaryl CoA and makes the product which is called tetrahydroxychalcone. So, this undergoes isomerization which makes naringenin. So, the naringenin is this one, or sometimes it is called naringenin chalcone. This can convert it into eriodictol. So, both eriodictol and naringenin comes under flavanones, and then subsequently by the flavonoid 3 hydroxylase F3H is flavonoid 3 hydroxylase, which makes this dihydroflavonol. These are the dihydroflavonol which are either dihydrocampherol or dihydroquercetin or dihydromycetine. So, these are called dihydroflavonols. So, the enzyme is flavonoid 3 hydroxylase. So, that basically as you see here at 3 position OH is added, and then what happens, this can converted into dihydroquercetin by F3 prime H. So, that means that another OH is added here, or F3 prime 5 prime H, what is there, one OH added here, another OH is added here. So, these structures maybe we will see in in the subsequent class, and then these dihydroflavonols can be converted to the flavonols. So, what are the flavonols? Quercetin, camphorol and myricetin, these are the flavonols. So, the enzyme is called flavonol synthase. So, this is this is the flavonol compounds, and so what is the differences here, that between dihydro and the camphorol, and the camphorol is here is one, this double bond, and then subsequently this converted into leucoanthocyanidin, which I have shown here. This is leucoanthocyanidin. So, which is by the reduction. So, reduction means that there is the reductase enzyme, and that is why it is called DFR, dihydroflavonol reductase. So, R stands for reductase, and then it makes leucoanthocyanidins, and then this will form the anthocyanin. Purposefully I am not discussing the anthocyanin in this class. So, here my purpose is to tell you the chalcone, flavanone, then dihydroflavonol, then flavonols and leucoanthocyanidins. So, leucoanthocyanidins taking part in the formation of other products also, that we will see. So, this is the leucoanthocyanidin. So, as I said, that plays one important role in the formation of anthocyanidins by anthocyanidin synthase, but as I said, this I will discuss in the next class. But the point to mention here, that this leucoanthocyanidin, by the action of leucoanthocyanidin reductase, which makes flavanthiols, which are epicatechins or epicatechin plus or minus epicatechins. So, flavanthiols, if you go here, the flavanthiols. So, here this is the flavanthiols, which are epicatechin or catechin. So, this is by the action of this reductase. Lipoanthocyanidin reductase, and then this subsequently joins together and form proanthocyanidin, which are big polymeric molecules, and also it comes under the condensed tannins. Okay. So, condensed tannins are basically formed by joining these flavonthiols in multiple numbers and form the condensed tannins. Whereas, in the right side what you see, this is basically the green tea. Green tea is basically a good source of polyphenol, which is basically dominated by epicatechin, epigallocatechin, epigallocatechin gallate, gallocatechin gallates. So, different. So, gallate is coming from the gallic acid, is coming from this secumate root, and catechin is coming from this root, and that joins, and it makes different complex polyphenols of green tea. So, but I am not going to discuss that, only I mention the names like epicatechin, epigallocatechin gallate, gallocatechin gallate, they are different compounds. So, once you make the extract, methanolic extract and run in HPLC, and if you have the standards, you will able to identify these compounds without any problem. So, the gallic acid, as I said, gallic acid can joins with a glucose, forms glucogallin. This slide I have shown in the previous classes, and that can also form the complex hydrolyzable tannins anyway. So, formation of flavonones. So, flavonones, okay, now let us go back again here. The flavonones is basically naringenin and eriodictol. So, naringenin and eriodictols, these are basically the flavonones. So, this naringenin or eriodictol can convert, can catalyze, can be used as a substrate for the enzyme called flavone synthase, and it makes either epigenin or luteolin. Epigenin and luteolin, these are basically the flavones by the action of flavone synthase, and it makes and you see the difference is pretty similar, only the hydroxy at R1 position makes luteolin, and these flavonones can be converted into flavonphorols by again a dihydroflavonol reductase. If I go back to this one, here you see there is a reductase here, flavonone by reductase it means flavonphorol. So, and flavonphorols which may be luteophorol or epiphorol, but this finally makes the flovaphen pigments as I have mentioned here, and this is basically the from flavonones, this different, either it produces the flavones or flavonphorols. Okay. And now come to the orones. So, flower color is dominated by mostly anthocyanin, but also carotenes, but also by orones, particularly in Canna species in monocot, Cannaceae family. So, their orones are prevalent, not only that, in some snapdragon also the yellow color is also dominated by orones. So, orones are basically the derivatives of chalcones only. As you see, this is orone. So, orone could be either yellow or red in color. So, orone is basically formed from tetrahydroxychalcone. So, tetrahydroxychalcone is the chalcone. So, that by the action of AS orosidin synthase. Orone is a group and the specific compound is called aureosidin. So, AS stands for aureosidin synthase, aureosidin synthase. So, it is here, and or the other root which operates in the legume, as I said that CHS and CHR will basically work together and makes isoliquiritigenin, so which is also coming under chalcone. So, tetrahydroxychalcone is a chalcone which takes part in the formation mostly for the flower color anthocyanidin, whether Another type of chalcone is called isoliquiditigenin, which is formed in the legumes, and that main role is formation of isoflavonoids. So, this once this isoliquiditigenin is formed, that also by the action of aureocidin synthase it makes aureocidin. So, this is called the aurones, and what we see that how the other isoflavones are formed. Okay. So, this is again a revision. So, chalcone synthase makes tetrahydroxychalcone, okay, which is taking part in the formation of naringenin and subsequently to anthocyanidins, whereas the another type of chalcone is called isoliquiditigenin, which is again a chalcone that plays important role in the formation of isoflavonoids. So, here naringenin is a flavonone, whereas another chalcone this isomerase which makes to liquiditigenin, which is basically a flavanone. Okay. So, chalcone then flavanone, right, correct, and then okay, then let us go to the next one. So, this is the reaction I have said in the previous class, no point of discussing. So, now the liquiditigenin is formed, and liquiditigenin by the subsequent enzymatic steps like IFS, O-methyl transferase and several, eventually it forms the compound. For example, if it is IFS, that isoflavone synthase, then it makes diadzine or genistein. So, diadzine is basically it is an isoflavonoid. So, it is a dietary phenolics, or through other route it produces genistein. This is also an isoflavonoid, and this pathway further moves and forms medicarpine. Medicarpines are also isoflavonoids, and so these are the complex molecules, but it is basically restricted to the legumes. So, legumes, in the legumes these pathways are operating. So, these are isoflavones and isoflavonoids. Only what you need to know that this isoflavonoid pathways are operating in legume. So, there the flavanone is basically liquiditigenin, not the naringenin. So, with this more or less I have covered the biosynthesis of total flavonoid group, which consists of flavanone, flavones, flavonols, isoflavonoids, isoflavones and aurone pigments. And also I mentioned about proanthocyanidins, which plays important role for the formation of condensed tannins. We also mentioned of flavone 3,4 diols, which makes the formation of flovapene. I also mentioned flavone 3-ols, which makes the which actually are nothing but the epicatechin, epicatechin gallets catechins. So, which are the components of green tea. So, this is in brief about the flavonoid group, and I will end the class here. In the next class, I will specifically talk about the anthocyanin biosynthetic pathway. With this, I end the class here. Thank you.